Welcome to CurveSetter MCAT Prep, the only MCAT prep company in the world staffed entirely by 99th plus percentile scorers. So today we're going to do another biology passage, and this time I picked one that seems kind of complicated. It's focused on acetylcholine receptors and things like that, and there are a couple of nice charts and stuff that will hopefully confuse you enough to justify me doing this video. So let's get right into it. When the neurotransmitter acetylcholine, ACH, is released from vesicles in the presynaptic terminals of neurons, it diffuses across synapses and stimulates specific postsynaptic receptors of adjacent neurons. Okay, so these are things that you should already know. Normally, most of the acetylcholine is degraded in the synapses by the enzyme acetylcholinesterase, which is synthesized by the same neurons that produce acetylcholine. Okay, so basically, as you know, you have acetylcholines packaged in these presynaptic terminals. You have calcium come in. These vesicles, they fuse with the presynaptic membrane and they eject the acetylcholine. And then the same neurons, the presynaptic neurons, also synthesize acetylcholinesterase, which will break down acetylcholine so that it stops acting on the acetylcholine receptor on the postsynaptic side. All right. Acetylcholine is abundant in brain regions that mediate learning and memory, and when present at relatively high levels, is also involved in pain relief. Our researchers used two drugs to study the role of acetylcholine in the learning and memory processes of rats. Drug X is an acetylcholinesterase inhibitor, and drug Y is a non-competitive blocker of postsynaptic acetylcholine receptors. Okay. So the second you read that, I want you to think of what the implications or potential implications of these drugs are. So drug X is an acetylcholinesterase inhibitor. Okay? So now I have to think, this is kind of like a double negative, right? So acetylcholinesterase is an enzyme that breaks down acetylcholine. So it stops acetylcholine from continuing to act on the postsynaptic receptors. So if you inhibit the protein that is breaking down acetylcholine, essentially what you're doing is you're increasing, so to speak, the amount of acetylcholine we have, okay? So drug X is akin to just adding more acetylcholine, right? And drug Y, it says, is a non-competitive blocker of postsynaptic acetylcholine receptors. So I don't know if it's really that important that it's non-competitive. Maybe it is. Essentially, it's saying that when something is non-competitive, it means that it's the inhibitor will inhibit regardless of how much of the substrate you have present. Okay, so they don't compete. The inhibitor will just bind at its own affinity for the receptor, and it'll just block it with no indication made to the actual substrate of the acetylcholine. Okay, so drug X is essentially increasing acetylcholine, and drug Y is decreasing acetylcholine's action. That's it. Groups of rats were maintained on different treatment regimens consisting of either a placebo, which is an inert substance, or a specific concentration of either drug X or drug Y. Okay, so the placebo is going to be the control, and then drug X and Y are the experimentals. Using mild foot shocks as a deterrent, so they're just doing fear conditioning, the researchers trained rats to avoid the avoidance conditioning, the darker compartment of a two-chamber training box. So there's some psychology in there as well. At the beginning of each learning session, rats were placed in a dark chamber and given 60 seconds before mild foot shocks were initiated. Additional shocks were administered every 60 seconds while any rats remained in the dark chamber. Okay, so essentially they're just teaching them to avoid the dark compartment. Okay, and this is a protocol that is done in labs all the time. You're just teaching them to avoid a certain spot. So let's just make a guess as to how these drugs are going to work. This is the best way to understand these passages is to make your own hypotheses and see if the passage confirms them or not. And that way you'll be really engaged and you'll have a good head as to what's going on in the passage. Okay, so we know that ACH is abundant in brain regions that mediate learning, so it's probably important for mediating learning. Okay, so if we administer drug X, maybe it'll learn quicker, it'll remember information for a longer time. So maybe you'll need less foot shocks to teach them. So if you apply drug X and then you do the foot shocks, maybe you only need like 3 foot shocks instead of 10 for them to learn that they shouldn't go to the dark spot. And probably if you give them drug Y, they'll just won't learn. Like no matter how many times you shock their foot, they won't learn. So I don't know if this is true, but this is my guess, and let's see if it works. And placebo should just be the normal, however many trials it usually takes for these rats to learn to not go to the dark side. Learning was scored by the number of trials required before the rats learned to avoid the shock 100% of the time. And memory was scored by the number of incorrect reentries into the dark chambers. Okay? So the number of trials that they needed in order to never go back to the darker side is considered to be learning, and memory is how many times they incorrectly reenter. Okay, so the placebo is just going to be used as a point of comparison. So that's going to be like the flat baseline. All right, and this line over here is for drug X. So remember, drug X is the one that inhibits acetylcholine esterase, so it should increase the amount of acetylcholine we have in the synapse. And so let's look at our axes real quick. It says the number of trials until 100% avoidance, and here's the dose. So at low to moderate doses of drug X, we see the number of trials required to avoid that dark side goes down significantly. Okay, so that's a good thing. That's what we want. So now the rat, it takes, so if you're using 10 milligrams per kilogram of weight of this, so it's just like a dose. It's controlling for how fat the rat is. So if it's a really fat rat, it's always going to need more. Okay, so if you at a dose of 10, it only needs around 10 trials to learn how to avoid versus the 50 that the normal placebo rat needs. So this is a really, really nice difference that we're seeing here. And we can see it's also dose dependent. So that's also a good thing, showing that it's another positive thing showing that the drug is actually effective. But what's interesting is that as you increase the dose even more, we actually see a flip. And I don't know if I've seen this that much in research. This is pretty interesting to me. And this is probably actually real data. So essentially, once you increase a dose after 10, which is like the maximally effective dose in this trial, we see that all of a sudden its learning just goes to shit. Excuse my language. But so if we're at 100, now it's as if you're not giving it a drug at all, right? It's acting just like the placebo. And if you give it 1,000, now it's really hard to teach it. I don't know why that works. I don't even know if that's something that's known or why that is the way that it is, but that's what we're going to go with. Okay, figure 2 says the effect of drug Y on shock avoidance. So remember it said that the number of trials was an operationalization for learning. So this is what they consider learning. And then the number of errors they called memory. Okay, so we should expect drug Y to block memory. Okay, so we should expect to see more errors when drug Y is administered, and let's see if that's true. Okay, so here's a placebo, here's the baseline number of errors. Of course, the placebo shouldn't be having any effect, and that's why as you increase the dose of the placebo, there shouldn't be any dose-dependent relationship with the placebo. Not supposed to be doing anything, right? It's probably like a sugar tablet or something. Or saline solution. Injection of just salt water. So we see that as we increase the dose of drug Y, they start to commit much more errors. Okay? And eventually that kind of drops off. It looks like it's approaching a plateau. So at some point, it'll just stop working. But it's pretty dose dependent in the first 100 milligrams per kilogram of body weight. Okay, and that's exactly what we expected. So the only thing that was kind of unexpected was the fact that at certain really high doses of drug X, we actually block learning, which is really interesting. I want to look that stuff up myself. But drug Y was pretty predictable. As you increase the dose of drug Y, we get more errors, which means that your memory is being blocked somehow. Okay. Let's go on to the questions. Acetylcholine is a transmitter in the striated voluntary muscular system. What effect would drug Y have on the functioning of this system? Okay, so in the neuromuscular junction, we know that acetylcholine obviously binds to the acetylcholine receptors on the sarcolemma or whatever, and that causes an influx of calcium, causes an efflux of calcium from the sarcoplasmic reticulum, which binds to like troponin, causes whatever, all those different things. The movement of those, the movement of the muscle fibers, the shortening of the muscle fibers, which causes muscle contraction. Okay, so all that stuff is what you have to bring up in your head. So what would Y do? Well, Y is blocking the normal communication of that receptor, right? So it's not letting acetylcholine bind to that receptor. So would it increase the synthesis of acetylcholine esterase? No. Why would it do that? This doesn't make any sense. It would increase the calcium influx into the T tubules. No, actually it would decrease it, right? Or not necessarily decrease it, you just get it less often. So no. It would block muscular contraction. Sounds pretty good to me. You don't have acetylcholine binding to the receptors anymore. How are you going to have contraction? And D, it would stimulate muscle. Okay, that's the opposite. So this one's obviously C. It's a pretty easy question. An underlying assumption of the researcher regarding the actions of drug X was that it, A, increased the amount of acetylcholine in the synapse, probably. B, affected learning by stimulating the autonomic nervous system. No. That's not an underlying assumption. We're talking about how drug X acts. Okay, so this is like too huge of a systems analysis. Like this is way too broad for the researcher to be able to assume that, right? It's insane. That's something that could be assessed, but that won't be assumed. C, act on the postsynaptic receptors. No, this acts on acetylcholine esterase, which is a protein that's released by the presynaptic neurons. It's not a receptor anyways. D, was synthesized in the same neurons as acetylcholine was. Yes, but that's not the action of drug. Oh, no, I'm sorry. Drug X is. I was thinking about acetylcholine esterase. Drug X is not produced by anything. It's an inhibitor of acetylcholine esterase. I don't think that's natural. Or not that it's not natural. Some animals might produce it, but it's not something that you would have in your own body. So we're gonna go with A, right? So it blocks acetylcholine. I mean, that's something that we talked about when we were reading the passage. It blocks acetylcholine esterase inhibitor. It is an acetylcholine esterase inhibitor, which means that it's blocking the breakdown of acetylcholine. So we should have more acetylcholine. So that's A. 152. If drug X and drug Y are given together, will the rate of learning most likely increase or decrease? Great question. Beautiful question. Okay. So there are a couple things that are important here. If we have drug Y, remember drug Y is a non-competitive inhibitor. So regardless of how much acetylcholine we have in the synapse, those receptors are going to be blocked. It's not dependent. It doesn't compete. It doesn't compete with the acetylcholine. So long as we add drug Y, it doesn't matter what we do with drug X. Drug X is not going to have any effect whatsoever. We can have unlimited amounts of acetylcholine, hypothetically, right? And those receptors will still be blocked. So adding drug X with drug Y should look exactly the same as having just drug Y. Okay? So, which means that the rate of learning should probably decrease, right? That's what Y does. You commit more errors and you don't learn as well. Okay, so A, increase because of increased production of acetylcholine caused by drug X will be overcome by the blocking effect of drug Y. Okay, so this is like a two-part thing where like there could be one part that's correct, another part that's incorrect, or vice versa. In this case, the rate of learning should decrease, but I think the reason is correct. because the increased production of acetylcholine caused by drug X will be overcome by the blocking drug. Yeah. So the second part is correct, but the first part's wrong. Okay, so we should find one that says decrease and has the same kind of reason as this one. B, increase because in general both drug X and drug Y act to increase acetylcholine production. That's just completely false, right? So not even drug X increases acetylcholine production. It just decreases breakdown. And drug Y binds to the receptor, that acetylcholine receptor, blocking it. So that's just completely out of the question. C, decrease because the excess acetylcholine produced by drug X will be degraded by drug Y before it can affect learning. That's wrong, right? Drug Y is not an acetylcholinesterase. It doesn't degrade acetylcholine. It just blocks its action on the receptors on the postsynaptic side. So that's out. D, decrease because although drug X will increase acetylcholine production, drug Y will prevent it from being taken up by postsynaptic receptors. Taken up is a weird word. It's supposed to be binding. Taken up implies that it's going inside, but that's the correct answer. It should decrease, and doesn't even matter if we have more acetylcholine, we have a non-competitive inhibitor, and it won't be able to do anything. It won't be able to bind to the receptor. The researcher discovered that increasing the concentration of drug X resulted in greater inhibition of the hydrolysis of acetylcholine by acetylcholinesterase. Okay, so that should be happening. However, this effect was counteracted by increasing the concentration of acetylcholine. These results indicate that drug X acts on acetylcholinesterase as what? Okay, so let's reread that. The researcher discovered that increasing the concentration of drug X resulted in greater inhibition of hydrolysis of acetylcholine by acetylcholinesterase. Okay, great. However, this effect was counteracted by increasing the concentration of acetylcholine. Okay, so what's going on here? So drug X is binding and inhibiting dose-dependently acetylcholinesterase, which makes sense, right? Just doing normal like inhibition on the protein. However, it's saying that this effect is counteracted by increasing the concentration of acetylcholine. Okay, so obviously both of them are binding. So acetylcholine also binds to acetylcholinesterase. That's how it gets broken down. But it's saying that the effect of drug X is being counteracted whenever we increase acetylcholine. So it's saying, what kind of inhibitor is this? The answer is competitive inhibitor. That's the type of inhibitor where it does it. So irreversible would mean that once it gets inhibited, it's done for. The enzyme is done. You can never fix it. You have to break it down. You can't remove the inhibition. So there's no indication of that. B, competitive, that's exactly what this is, right? So if the substrate is now competing with the inhibitor and you can just outcompete it. So if we add enough acetylcholine where the effect of the inhibitor is just completely eliminated, then that's definition competitive inhibition. Non-competitive inhibition, we said before, is the case in which it's not dependent on the substrate concentration. So that adding more acetylcholine would not affect the ability of the drug to block acetylcholinesterase. And that's what it originally said it was. I'm sorry. It didn't say that. It said drug Y is non-competitive. I apologize for that. So yeah, but that's not what's happening, right? Because it's saying that we increase the concentration of acetylcholine, that it counteracts drug X's ability to block acetylcholinesterase. Okay, that's not what non-competitive is. And then mixed competitive, non-competitive inhibitor. So yeah, so mixed competition has to do with when to what complex the inhibitor binds to with greater affinity. So it can either bind. So an inhibitor can either bind to the enzyme complex itself or to the enzyme substrate complex In this case, there is no mixed inhibition going on. It's simply just competitive inhibition. So we're going to go with B. Okay. Based on the experiment in this passage, the best overall description of the effect of acetylcholine on rat learning and memory is that moderate increases in acetylcholine utilization appear to disrupt. No. It's the opposite, right? We like that. It takes less trials for it to learn. Okay. Moderate increases in acetylcholine utilization appear to improve memory. That sounds about right. Blockage of acetylcholine receptors appear to improve. No, absolutely not. Doesn't improve anything. Makes it worse. More errors. Blockage of acetylcholine esterase appears to improve learning at low doses. So this is actually, there's none of this, right? You're seeing errors increase no matter what. It's not like they decrease first and then increase or something, or decrease, or increase and decrease. So the answer is B. That's referring to this figure right here. So at low levels, moderate increases, we see a nice increase in learning, right? Less errors. These last two have to do with figure 2, and in figure 2, it's obvious from the very outset that no matter how much drug Y you add, you're gonna mess up learning and memory. Okay, you add a little bit, get a bunch of errors. You add more, you get a lot more errors. 155. The fastest way to counteract the effects of drug Y would most likely be to what? Increase its rate of metabolism and elimination of the body. Okay, maybe administer increasing amounts of drug X. So remember that drug Y is a non-competitive inhibitor. Adding more acetylcholine, which drug X kind of does, doesn't do anything. Administer a competitive receptor stimulator. Again, that's the same thing as adding more acetylcholine. Drug Y acts non-competitively, so adding more of the receptor stimulator is not going to do anything. Administer another non-competitive acetylcholine receptor blocker that acts at the same site as drug Y. So that's kind of a funny choice. All that would do is like, now that drug is competing with drug Y for the spot, but they're both still inhibiting the enzyme. So while it will like counteract like drug Y's ability to like bind, it won't counteract its effects. That's kind of the key word. You're counteracting the effects of drug Y. The effect of drug Y is to block the receptor. So if we add another blocker that might like replace drug Y, it's still a blocker and it's still screwing up learning and memory. So the best way to do it is just to get rid of the receptor and just put in a new one that doesn't have that blocker bound to it. Okay. An autosomal recessive non-lethal mutation deletion of the acetylcholine receptor gene occurs in some rats. Okay, so we just don't have the receptor in some of them. Autosomal recessive non-lethal mutation. Interesting how it's not lethal. It's probably always just a subset. Which of the following organelles will most likely be abnormal in the rats having this mutation? Microtubules, ribosomes, endoplasmic reticulum, plasma membrane. Okay, so you tell me, where do we find acetylcholine receptors? Because that's where would it be abnormal, right? Do we find them in microtubules? No. Ribosomes? No. Endoplasmic reticulum? No. Plasma membrane? Yes. That's where they reside. That's what receptors do. They're on the plasma membrane. So the answer is D. Alright, so I hope you enjoyed this video. If you'd like to get tutoring for the MCAT, our company has a great program available for you at amazing rates. Again, all of our tutors scored in at least the 99th percentile on the MCAT, so a 522 or higher. A lot of our tutors actually got 100th percentile scores, and we all attend top medical school, so we're here to help you. You can visit us at mcatprep528.com. Thanks.